Do you know how you get aminoglycoside resistance? Well, bacteria like Pseudomonas aeruginosa are going to neutralize your aminoglycosides by chemically modifying them. Usually how this happens is enzymatic acetylation, and that's going to block your ribosomal binding. So aminoglycoside is going to be acetylation. It prevents the 30S ribosomal binding, reducing drug efficacy. This is plasmid mediated. It's not due to chromosomal. Seen in gram-negative nosocomial bugs like Pseudomonas aeruginosa. And your beta-lactams, like your penicillin cephalosporins, are going to use beta-lactamases, not acetylation. So here's our practice question to see if we understand this. 79-year-old man has a chronic indwelling Foley catheter and is admitted for fever and altered status. Urine shows Enterobacter colicae, and this is the isolate is resistant to gentamicin due to the presence of the plasmid-encoded enzyme that transfers acetyl groups. So what's the result? It's going to be an impaired 30S ribosomal subunit interaction. So gentamicin, tobramycin, any of the mycins, like your aminoglycosides, are going to bind to the 16S RNA of the 30S ribosome, and that leads to mRNA misreading and bacterial death. That's how it works, right? It's like, oh, you can't read. It's like you ever play basketball with somebody, and they're like, ooh, in your face, can't see. That's exactly what your aminoglycosides gentamicin is going to be doing. It binds to that 16S and the 30S of the 30S, and it leads to a misreading and bacterial death. So if you modify and you acetyltransferase this binding site, then the drug is going to be ineffective. So let's talk about some other answers. Peptidoglycan inhibition. Peptidoglycan, that's beta-lactam, so like your penicillin cephalosporins, is mediated by your lactamases, not acetylation. DNA gyrase, that's the MOA of the fluoroquinolones, so that'll come from mutations or efflux pumps. Enhanced permeability, that would increase the sensitivity, so no. 23S, that's the mechanism of resistance for your macrolides, not your aminoglycosides. Those guys are the 16s. So yeah, aminoglycosides are silenced by bacterial enzymes that acetylate drugs. Why? Because it blocks the binding of the 30S ribosome. That mechanism spreads via plasmids, and it's a super common form of resistance in gram-neg pathogens like Pseudomonas. So yeah, mutations in like the efflux and DNA gyrase and topoisomerase, that can definitely do it. But here we're talking specifically about aminoglycosides. It's going to be that 30S guy through acetylation. All right, hopefully that was helpful. If it was, consider liking and subscribing, and we'll see you guys in the next video.